Hi there, my name is Hannah Gannon, and today I have my sixth interactive case study with you all. So we are going to be discussing tubulin, which is a protein that's actually in all the cells of our body. It plays a really important role, especially when it comes to the cytoskeleton and cell division or mitosis. And we're going to also look at how it can be used and targeted specifically in different cancer therapies. So to begin, I'm just going to give a brief overview over tubulin, its function, and how it's really crucial specifically for cell division. So to begin, I just want to go over a little bit about what mitosis is. So mitosis is a process. It's extremely important and it just involves cellular division. You're going to go from one cell and you're gonna make two identical cells essentially. And that's pretty much the briefest overview I can give in terms of mitosis, but obviously this is a really crucial process, and it's something that occurs throughout our entire lifetime. Our cells are constantly dividing as well as dying, and these processes are really regulated, and that's something that's really key here. Now, something that's really crucial that happens during mitosis specifically is we have chromosomes and they're going to be moving and segregating also. And this is actually controlled specifically by what's called the mitotic spindle. Now, the mitotic spindle is actually composed of what's called microtubules. And then if we want to go, a little bit further, you can say that the microtubules are composed of what's called alpha and beta tubulin heterodimes. So going back forward a little bit, so tubulin heterodimes make up microtubules, which make up or lead to the mitotic spindle. Now, something that you can see in this picture here that I have is we have like the two different types of tubulin. They create this dimer, and then this creates this microtubule, which is really crucial, specifically at a certain part of mitosis, which we will go over. So this plays a role, like I said, with mitotic spindle, which essentially has the role of, well, one of the most essential roles of this is actually going to be separating the chromosomes. The chromosomes line up on the kind of like the equatorial plate, and it's actually going to separate those chromosomes so that they can go into the daughter cells. So next I am going to just go over a little bit more in depth of this process. So like I said, microtubules are going to attach to the chromosomes, and then they're gonna be aligned in the center of the cells, and the tubules are actually gonna shorten, and they're gonna move to then the chromosomes are gonna be moved to opposite poles, hence moving to to the two daughter cells. And so this is just like, so this is just like a very essential process, and it's really important that this is really highly regulated, just because you don't want, like two chromosomes to go in one daughter cell and like the other chromosome or the other cell to get no chromosomes. So you really want this process to be like highly regulated, obviously, for apparent reasons. Now, one thing that I did want to mention though is that so like I said, so tubulin creates these, long chains of microtubules, and that's just really going to make up the skeletal structure for cells. And one really interesting thing that I found about tubulin is just how flexible it is, and this is really crucial when it comes to the formation, shifting of microtubules. And so I think that that's just a really great example of how structure leads to function, and so understanding like why it's flexible and like how that function can be carried out specifically in the process of mitosis, I think is really interesting. Now that we kind of understand, how what tubulin is specifically, and how, this leads to like bigger organizational structures within the cell, such as microtubules, the mitotic spindle, I wanted to go over how this can be, targeted in different types of cancer therapy. But before I do that, I do want to just show a brief overview of kind of like big picture wise what we had discussed. So like, let's say we're in the middle of, cellular division here, or mitosis. We've got these sister chromatids, and as you can see, they are beginning to be separated. They're going towards opposite poles here. And then we have these microtubule structures all around here. So you can just see like how important these structures are. They make up a major component of mitosis and how it's able to be really carried out in the cell. Now, in terms of how Oncogin, which is Oncovin, which is going to be the drug we were discussing, we're going to kind of talk about how it specifically targets tubulin, which is the protein that actually makes up those microtubules. So before I go into that, I just want to kind of talk a little bit about like what cancer is. So I guess the briefest way I could kind of describe it as just like uncontrolled cell proliferation. There's almost just like no breaks, cells constantly divide, hence you get, tumors. And, this, it can be caused by a number of things. Some of the most common reasons would be either you're having like constant cell division or uncontrolled, like I said, proliferation and mitosis, or there's just no apoptosis, so cells aren't like being programmed to die appropriately. And so there's like many causes to cancer, obviously. Now, one thing that I found when I was kind of researching this topic, though, was that there's actually evidence that tubulin and microtubules regulate certain functions in cancer. So there's actually what's called tubulin binding drugs, which would be Oncovin, for example, and they are able to kill cancerous cells by actually inhibiting the microtubule properties, and this allows for the DNA segregation to like not occur because those microtubules are so essential for the segregation of the DNA, and therefore cell division cannot be continued at that point. So in terms of like a big picture of like how this drug works in the body, it binds to the tubulin protein, and so the tubulin protein is unable to contribute to the separation of the chromosomes and this is what's called metaphase of mitosis, and the cell ends up undergoing apoptosis or programmed cell death as a result. So in terms of how it binds relatively, or in terms of how Oncovin binds to tubulin, it binds relatively quickly as well as in a reversible manner. And it's really kind of fascinating because this actually results in a conformational change in the tubulin protein, and it creates these paracrystalline aggregates, and as a result, tubulin dimers are not able to be created and available to create microtubules, and as a result of not having microtubules, or the microtubules actually like shrink and disassemble, and eventually you're not going to have the available microtubules that you need to segregate the DNA for mitosis. Another thing that I did want to mention as well is how, and this is something that actually has to do with like a lot of different cancer therapies, is that you're not able to target certain cancer cells. So a lot of times any cells that are proliferating a lot or rapidly dividing, they are going to be targeted whether or not they are cancerous. And so that's something that just needs to be considered from an administration point, is just being able to administer and being really meticulous with like what you're administering in terms of like dosage and everything like that, because you're not able to target certain cancer cells. So all of the cells are going to be impacted in this way, and you're going to impact the mitosis or cellular division that's going on in the human body. So overview, big picture again. Oncovin, which is a drug that's used for cancer therapy, it's going to inhibit the polymerization of tubulin subunits, so microtubules are not able to form, and because of that, you're not able to create that separation of DNA or sister chromatids that is so necessary in terms of mitosis and cell division, so the cell's not able to divide, and as a result, apoptosis occurs. So, and like I said, it actually ultimately results in the destruction of mitotic spindles. And yeah, the dividing cells are not able to, or the cells are not able to divide anymore. And then just before I end, I just wanted to give credit to my sources that I used in terms of getting all this information, as well as the pictures that I used. And so just to reiterate, tubulin is a protein that is in our cells, and it's essential, especially in terms of like the cytoskeleton structure, and forms like the basis of really the mitotic spindle. And all of this is really necessary for the process of mitosis, and this is something that the different cancer therapies can really target at and take advantage of. One specifically is Oncovin, and that's going to bind to tubulin, and as a result, you're going to really inhibit that segregation of DNA, so the cell's not able to continue dividing because you're not able to, you know, continue mitosis in that way because you impaired the microtubules and the mitotic spindle by really looking at the tubulin protein there. So I hope that this makes sense, and thank you for watching this video, and have a wonderful night.